Hello and welcome to Conversations on AFM. I'm Dr. Elizabeth Kugler, a science communicator and your host of today's third conversation, Insights from a Scientist, the Nanomechanics of Living Cells. To talk with us about one of the most interesting cell types, which are endothelial cells, is our guest, Professor Kristina Kusche-Vihrog. She is the director of the Lübeck Institute of Physiology, on the board of the German Hypertension League, and she received the German Society of Nephrology Award for research into hypertension in 2018. Her research focuses in particular on the influence of inflammatory processes on endothelial dysfunction as a precursor to the development of cardiovascular diseases and hypertension. She is here with us today to talk about AFM, and she uses AFM to understand the nanomechanics of cellular surfaces and the biophysical methods in basic medical research and diagnosis. So thank you so much for being with us here today, Professor Kusche-Vihrog.
Yes, thank you very much for inviting me. I'm really happy to do this today.
We're really excited to hear from you today about your work and especially about endothelial cells. So for our listeners who might not know your work and endothelial cells, we thought maybe it would be great if you could introduce to us what you're researching on and what role endothelial cells play.
Actually, when I started with my research project as a postdoc, I started with epithelial cells. They are also very nice, but then after a while, we recognized that the endothelial cells, which are lining the blood vessels, the inside of the blood vessels, have a high impact on the vascular system, and they also have a lot of ion channels in their plasma membranes, and they are highly flexible, so they can react to the forces of the blood, and they have a very cool position in the inside of the blood vessel because they can sense everything which comes with the blood. And the most important thing is that they can react. So they sense, for example, vasodilating factors, and then they give signals to the vascular smooth muscle cells which are surrounding them. And so they have a very important job in our vascular system. So, and this is why we started to have a closer look on the mechanical properties of these cells. Yes, because of their position inside of the vasculature, like a layer of the blood vessel. So as I said, they sense everything, and the most important thing is they sense also the forces of the blood. And so they can be deformed by these blood forces, by the streaming blood. And when they are deformed, they react in a certain manner, like they give vasodilating factors or they have vasoconstrictory factors, and so they are big regulators in the vasculature. And so we recognize that this behavior of the cells is absolutely important in terms of physiology, but also in pathophysiology. So when they behave like they have to behave, so they are flexible and they are deformable and they can switch between different mechanical states. I think we talk about later about these different states. So they have a normal and healthy behavior. And so when something happens in the body, like disease, inflammation, they change their behavior, they change their mechanical properties, and then we have so-called endothelial dysfunction.
Maybe you could talk a little bit about what cell stiffness means for kind of a body and like the mechanical properties, like how we measure them and what that means for like the surrounding tissue.
Yes, that's a very important point in endothelial function and dysfunction. So we have this underneath the plasma membrane, we have an enzyme. It's the eNOS, the endothelial NO synthase. So this is known for many, many years. And when the cells are deformable, when they are very soft and deformable, so they have a cytoskeleton which is more in a deep polymerized state. I can explain this later maybe about the cytoskeleton thing. So when they are deformable, this enzyme is activated. And so the streaming blood by itself can activate this enzyme. And so when this enzyme is activated, we have this typical cascade, and we need arginine and all these known things. And then we have a production of NO in the cell. And the endothelial cells, they produce then this NO as an answer, as a response to this deformation from the bloodstream. And this nitric oxide is like a gas. It's the half-time is very, very short, but it can diffuse to the vascular smooth muscle cells, and then they relax. And so we have an increase in cell diameter or in vessel diameter. And so we have a decrease in blood pressure. And that's an important mechanism. So when we stand up, for example, it's this autostase reaction, then we have the constriction, but then we need other situations where we need more vasodilation. And this is all connected to the mechanical surface. So and the thing is that I'm talking about only the surface of the cell. To be clear, it's only the 150 to 200 nanometers on top of the cell. So it's the plasma membrane, so it's the glycocalyx, the plasma membrane, and the layer underneath the plasma membrane, the so-called cytoskeleton. It's only 150 to 200 nanometers. And this is the highly dynamic outer layer of the cells, which can react to this blood forces. And there we have the release of the nitric oxide. And this is very potent vasodilator, for example.
Yeah, you already mentioned now the glycocalyx. Maybe you could talk with us about, what is it and how do you measure it? Is it, for example, cell culture? And how do you then, for example, use AFM to measure the glycocalyx? What role does it have in endothelial cells and cell stiffness?
Yeah, the glycocalyx is the best structure I ever studied. So it's really, really cool. And I'm a big glycocalyx fan because actually the most important thing about endothelial function is the integrity of the glycocalyx. So it's really high, but it's really hard to measure. But with the AFM, we have the perfect instrument to measure the glycocalyx. So we touch with a spherical tip. We use very small tips, like 1 micrometer in diameter, and very, very low loading force in the range of 0.5 nanonewton. So it's very soft because the structure is so flexible and vulnerable. So we have they cannot use high loading forces. Then we just touch the surface of the cell and then we indent the cell. So we measure the force we need to indent the cell for a given distance, like the Hooke's law. And so when we first indent the cell, the first nanometers, then this is the glycocalyx. And then after, so when we indent more, then we touch the real surface of the endothelial cell, then it's the plasma membrane. And when we analyze the AFM curves, so we have different slopes, and it took us years and years to analyze these slopes, and we used enzymes like heparinase to remove the glycocalyx enzymatically. And so we found out that we really can quantify the height and the mechanical property of the glycocalyx and also the mechanical property of the cortex of the cell, which is the layer underneath the glycocalyx. And yeah, the glycocalyx is like a mesh, so it's built of many factors or glycoproteins, proteoglycans. Heparan sulfate is a very important compound of the glycocalyx. There are some receptors inside the glycocalyx, and it's like a shield. It's like a vascular protective structure on top of the cells. So, and it's really hard to quantify this, but I must say with the AFM, we have a really nice instrument to do this.
It is a wonderful structure. I think that's the thing, like it's often not considered as a kind of important structure per se. People just like think it's part of the cell as such, but it doesn't really have a role on its own.
Yeah, but here look at this classical leukocyte adhesion cascade, which you can find in every textbook, and the glycocalyx is completely neglected. So they are the pure cells with a naked surface, and the first thing that the leukocytes meet on the surface is the glycocalyx. And so we have the conversation between the leukocyte glycocalyx and the glycocase of the endothelial cell. And so these structures, they touch, they interact, and then something happens. The glycocalyx is removed by action of the leukocyte, and then the way is free for adhesion and transmigration. And this is still neglected in all textbooks.
And one other question I had was like, is it in cell culture that you do your experiments, or do you kind of what type of cells is it that you really work with?
So yes, we have different models. So actually UVECs are our gold standard. So we freshly isolate them from the umbilical vein here from the hospital at the University of Lübeck. And so we isolate the UVECs. So it's, we have them in the lab every day. And in German we would say it's our Haustier. Would you say it's a pet?
Yeah, it's a pet.
Yeah.
Yes.
So, but we have, but they don't like to be transfected. So when we use, for example, CRISPR-Cas to knock out specific channels or whatever in the cell, so we use this EAHU cells. It's a cell line. But I mostly prefer to work ex vivo. So we use blood vessels from mice or patients and split open them. And so we have the endothelial cells facing upwards. And so we can measure, we do our measurements on these endothelial cells in ex vivo blood vessels. And this is perfect because so we have patient vessels, or we can use different mouse models, transgenic mice, and so on. And this is a model we worked really hard to get this model, but now it's working so nice, and we are so happy with it.
That's amazing. I think it's always like until you got the workflow and the experiments and the protocols, it's such hard work. But when it works, it's so great.
It must be said that when you try to culture the endothelial cells, the glycocalyx needs time. And so this is often a problem that other labs, they want to they want to be fast, and so they culture their endothelial cells only 2 days or something like this. And this is not long enough for a proper glycocalyx. So the cells in culture and also ex vivo preparation patches, they need time. So when you stress the endothelial cells after this isolation process, for example, then it takes at least 3 to 4 times until the glycocalyx is recovered, and then you can measure it. And of course, it's not that high as in vivo, but then we have we bought this glycocheck camera. Can I say this? It's wonderful. So we can also measure the glycocalyx on tongue capillaries from patients.
That's amazing. So it sounds really like your research is so close to the patients, and obviously you work with the hospital and you get the samples. So it's really having a direct impact and relationship to the hospital and patients, which is always great to hear. Which also kind of brings me to the next kind of question that I had, namely that obviously in a lot of vascular diseases, such as, for example, high blood pressure, which is probably the most well-known disease because about 50% of adults are thought to have hypertension according to the CDC. So in hypertension, we have cell stiffness being increased of the blood vessels, and obviously this can lead to inflammation and kind of secondary issues. And your work directly works on cell stiffness. So how do you see your work really directly translating to medical research?
Yeah, that's a very good question. So basically, the endothelial stiffness can be directly correlated to arterial stiffness. And when we found out this, we were really, really happy. And so this was in a project where we worked with a pediatric cohort. So it was a chronic kidney disease cohort. So children suffering from chronic kidney disease. And so we measured, it's another model. So we used the UVECs and incubated the UVECs with patient serum. So we have all these pathologic compounds in the serum and incubate standardized UVECs. And here we found that these factors in the uremic sera completely damage the glycocalyx, stiffens the cell cortex, and this stiff cell cortex could be correlated to arterial stiffness measured as pulse wave velocity. So what we found on the single cell level is also true for, let's say, great parts of the arterial system. So we have local reactions, so where we have local vasoconstriction, for example. But in all these inflammatory disease, I'm convinced that the behavior of the endothelial cells has an impact on the complete arterial system.
Yeah, I absolutely agree. I mean, it's also like I was reading about your work, obviously, and one study that you're working on is systemic sclerosis. And I was exactly thinking about that, where you worked on antibodies and autoantibodies, and it's obviously like a multi-system connective tissue disease impacting the whole body. So I thought maybe you can share a little bit about your research on systemic sclerosis and cell stiffness, and again, maybe the kind of more global components that you think have a role in this.
Yeah, that's an ongoing project. So we have preliminary data, but I can, of course, talk a little bit about autoantibodies. It's the same is true for SARS-CoV-2, COVID. It's more or less the same. So we have inflammatory disease, and the body produces so we have always autoantibodies. And when something happens, it's like a kind of black box. The autoantibodies, they bind against, for example, G protein coupled receptors like angiotensin II receptor. And the binding of these autoantibodies against this G protein coupled receptors, they completely change the endothelial surface. So we find that the glycocalyx is damaged. So the height is flattened completely. It's a shedding of the glycocalyx, and we have a stiffer cortex. And so the working hypothesis is that in disease like systemic sclerosis or other inflammatory disease, we have the self-reaction of the body, and when these autoantibodies bind to these receptors, we have specific cascades, and leading to inflammatory processes. Because these receptors are all linked to the cortical cytoskeleton. This is what you also have to talk about. We have underneath the plasma membrane, there is a specific cytoskeleton. It's not the stress fibers inside the cell. It's a very narrow compartment. It's, let's say 200 nanometers, and this is called cortical cytoskeleton. And this is actin, myosin mesh, like always, and we have linker proteins. And many of these structures, like receptors, also the glycocalyx, of course, via syndecan or glypican, they are linked to actin. And there are two options. So either we have this shedding of the glycocalyx, or we have maybe a cortical stiffening, which leads then to a polymerization of the cortical actin. And then so we have an intrinsic signal which leads to a collapse of the glycocalyx. So we have different ways to damage the glycocalyx and to damage this complete compartment of this outer shell of the endothelial cell. It's complicated.
I love that. No, that was beautiful. So, for example, the actin cytoskeleton, right? Would you, for example, then use like a chemical to disrupt the actin cytoskeleton and see how the glycocalyx kind of dissolves, or how do you work then with the actin cytoskeleton in this?
Yes, we did it. We can manipulate the actin cytoskeleton, and so it is important when you do this fluorescence microscopy to focus really on the cortical cytoskeleton and not on these stress fibers. For my research, I don't want to see them, so I only focus on this cortical actin. It's good to have a confocal microscope then. And of course, we can manipulate this cortical cytoskeleton. We can use just flavonoid to polymerize it, or we can use cytokeratin D to depolymerize this actin mesh. And we can, when we manipulate this actin fibers underneath the plasma membrane, we have reactions from the glycocalyx. So a stiff cortex leads to a collapse of the glycocalyx, and a soft cortex gives us a wonderful, upright and proper glycocalyx. That's the link.
Moving on, I kind of want to start asking you a little bit about the future. So, for example, in your opinion, like we talked a little bit about the sample preparation and getting the protocols ready is probably one of the most difficult challenges. But I guess there's also other challenges with AFM and using the technology in the lab. Is that like a technical challenge, do you think? Or like what other challenges with respect to AFM are you seeing in your research?
So when I look back to my first AFM measurements, they were really hard. But now the modern AFMs, they are so nice. They're very, very, very nice to use at the moment. I think it's more the cells. So it's absolutely imperative to handle the cells in a physiological way. And I think this is really the difficult thing. We have living cells, and only a living cell in a perfect surrounding acts like a living cell. So pH, salt content, and everything must be perfect, and the time of growth must be perfect. And so it's very important to study the morphology and the behavior of the endothelial cells prior to the experiment. And you need a lot of experience to see in the cell culture, oh, now the cells are in a really good shape, now that we can go to the experiment.
So where do you think the future of AFM is going, and what role does it have for you as scientists?
So from my point of view, the AFM is one of the best methods to quantify cell mechanics in a really reliable manner. So I'm really a big AFM fan. And it is so perfect to see online how a living cell reacts when I apply substances. And I really would wish that we can standardize the measurements in this way that we can have the AFM as a diagnostic tool. This would be so nice. So we have, for example, in chronic kidney disease, the patients, they have biopsies in the kidney. This hurts and it's not very nice. But from my research, from my group's research, I know that when we use patient sera and incubate standardized UVACs with it, we can differentiate between CKD stage 3, 4, and 5. So we have significant differences in cortical stiffness and glycocalyx height by only using sera from patients. And this is non-invasive, and it took time to create all these force curves, but it would be very, very nice to integrate the AFM as a diagnostic tool in translational projects and also in the clinics.
Yeah, it gives a completely new dimension to disease, doesn't it? If you could say, I take a little biopsy, measure stiffness, get insights into glycocalyx, actin, cell health, etc. I think that would be, like you say, really cool to have it as a diagnostic and like early on marker instead of like looking later on into cells. That's beautiful. Yeah. Are we far from that? Or where's the status?
No, we are not far from it. So in our projects, we can do this at the moment. So because endothelial dysfunction, what we measure is endothelial dysfunction. A stiff cell is a dysfunctional cell, and the damaged glycocalyx is also a hallmark for a dysfunctional cell. And I'm convinced that we see it every day in the lab when we measure the cells. And at the moment, in some projects, we could do diagnostic, but it's not in the mind of the clinicians at the moment, because it's a huge machine and maybe a bit scaring. I don't know. It's too exotic at the moment, but I try to convince many groups to try it. Give us our sera, and I can give you details about endothelial dysfunction. And here in Lübeck, they are pretty open. It's very nice.
That's amazing. It's so wonderful to hear how really science has an impact on daily lives, and we learn more about biology, but also how it directly translates into clinical research and how you work with clinicians, which is always really exciting to hear. We really want to thank you so much for your time, for joining us today. You have really inspired us and hopefully many of our listeners to now look into your work as well as into AFM. Thank you so much for your time. It was really fantastic to hear more about your work.
Thank you very much.
So listeners, if you have not already done so, be sure to listen to episode 1, where we talk to Emeritus Professor Dr. Mervyn Miles about the history of AFM, and in episode 2, where we have a fascinating conversation with Dr. Andre Koenig, where we talk with him about the applications of bio-AFM in the life sciences. Thank you for listening.
This podcast was sponsored by Bruker Bio-AFM, part of Bruker Nanosurface and Metrology Division. Bruker is a manufacturer of scientific instruments for life sciences and materials research, as well as for industrial and applied analysis. If you'd like to learn more, please visit www.bruker.com/bioafm. This podcast was produced by Keep It Science.